everyone, let's practice some MCQs on beta oxidation of fatty acids. The first MCQ is beta oxidation of fatty acids requires all the following coenzymes except A. CoA, B. FAD, C. NAD, D. NADP. The right option is the coenzymes for the beta oxidation of fatty acids are all of these options CoA, FAD and NAD except NADPH. NADP is not required in the oxidative process of fatty acids. As shown here is the pathway of beta oxidation pathway, in which if you see here after the activation of fatty acids, after the activation of long chain fatty acids, it is a 4-step reactions in which 4 enzymes act. So in this 4-step enzymatic reaction, at the first step NAD is required, FAD is required, and in the third step NAD is required, and at the fourth step CoA is required. So in this whole pathway, FAD, NAD, CoA, all these 3 are required in the beta oxidation of fatty acids, while NADP is not utilized for the beta oxidation of fatty acids. Question 2. Which of the following can be oxidized by beta oxidation pathway? A. Saturated fatty acids, B. Monosaturated fatty acids, C. Polyunsaturated fatty acids, D. All of these. So the right option is D. All of these. As the beta oxidation pathway, all these can be oxidized by this pathway. As shown here is the beta oxidation pathway, in which the first step as shown here is the activation of long chain fatty acids, and then after that there is a 4-step reaction in which 4 enzymes are required. At the first reaction FAD is required, second step is the hydration, the third is again dehydrogenation reaction, and the fourth one in which there is the cleavage. So all these fatty acids are oxidized by this beta oxidation pathway. The examples for these fatty acids are saturated fatty acids. Examples are stearic acid, which is 18 carbon fatty acid, palmitic acid, which is 16 carbon fatty acid, lauric acid, which is 12 carbon molecule. Examples of the monounsaturated fatty acids are oleic acid 18 carbons, palmitoleic acid 16 carbons. While polyunsaturated fatty acids examples are linoleic acid, which is 18 carbons, alpha-linoleic acid 18 carbons, and arachidonic acid, which is 20 carbon molecule. All these saturated, monounsaturated and polyunsaturated fatty acids, all these are oxidized by beta oxidation pathway. But for the monounsaturated fatty acids and polyunsaturated fatty acids, before going into the beta oxidation pathway, there are some additional steps and reactions before they enter into the beta oxidation pathway. And similarly, the polyunsaturated fatty acids, before they start the beta oxidation pathway, there are some additional steps. CoA is formed in the oxidation of A. Monounsaturated fatty acids, B. Polyunsaturated fatty acids, C. Fatty acids with odd number of carbon atoms, D. None of these. As propenyl-CoA is a 3-carbon molecule, so it is formed when the fatty acids with odd number of carbon atoms undergo oxidation. So the right option is C. How? Propenyl-CoA, as you can see here, this propenyl-CoA is a 3-carbon molecule. So when the fatty acids are with odd number, then the end one of acid, instead of formation of acetyl-CoA, we get the propenyl-CoA. As in beta oxidation, the fatty acids undergoes repeated cycles of beta oxidation where 2 carbon units are cleaved off in the form of acetyl-CoA. As in beta oxidation, when the carbon atoms are removed in each reaction, their 2 carbons are removed in the form of acetyl-CoA. So it is repeated until all the carbon units of the fatty acids are oxidized. But in case of this odd fatty acids, In the last round of beta oxidation for an odd chain fatty acids, 3 carbon unit propenyl-CoA is produced instead of the usual 2 carbon acetyl-CoA. As in beta oxidation of fatty acids for even number of carbon atoms, for each round 2 carbons are removed in the form of acetyl-CoA. But when they are odd, instead of 2 we are left with 3, which is the propenyl-CoA. So propenyl-CoA is formed when the fatty acids with odd number of carbon atoms, they undergo the beta oxidation of fatty acids. All the following statements about carnitine are true except A. It can be synthesized in the human body. B. It can be synthesized from methionine and lysine. C. It is required for the transport of short chain fatty acids into mitochondria. D. Its deficiency can occur due to hemodialysis. So they have asked about all the options are right except one. So option A, it can be synthesized in the human body. Yes, this is true. It can be synthesized from methionine and lysine. Yes, this option is also correct. C, it is required for transport of short chain fatty acids into mitochondria. No, this is wrong. So the right option is C, as the carnitine is required for the transport of long chain fatty acids into mitochondria. It is not the short chain fatty acids of mitochondria into the mitochondria. D, its deficiency can occur due to hemodialysis. Yes, this is true. In hemodialysis, as carnitine are very small in size, so during hemodialysis it may be lost, and the loss can cause the deficiency. So carnitine, as they are involved in the transport of long chain fatty acid from outside from the cytoplasm, from the cytosol to the inside of the mitochondria. All of the following can be oxidized by beta oxidation except A. Palmitic acid, B. Phytanic acid, C. Linoleic acid, D. Fatty acids having an odd number of carbon atoms. So the right option is phytanic acid. So palmitic acid, linoleic acid and fatty acids having an odd number of carbon atoms, all of these can be oxidized by beta oxidation, while phytanic acid is the fatty acid which cannot be oxidized by beta oxidation. Instead of that, phytanic acid are oxidized by alpha oxidation. Phytanic acid cannot be oxidized by beta oxidation because of a methyl group at 3 position on the fatty acid chain. Instead, phytanic acid is oxidized by alpha oxidation. So phytanic acid, as shown here, this is the structure of the phytanic acid. It has the methyl group at the 3rd carbon. So that's why at every 3rd carbon there would be the methyl carbon. So that's why it does not undergo the beta oxidation. Instead of that, it will break down the fatty acid on alpha carbon. So at every alpha carbon, it will undergo the alpha oxidation pathway. So that's why phytanic acid, it cannot undergo beta oxidation. Instead of that, it will follow the alpha oxidation. So it will break the carbon chain and fatty acids at alpha carbon. Lipids are stored in the body mainly in the form of A. Phospholipids, B. Glycolipids, C. Triglycerides, D. Fatty acids. So the right option is lipids are mainly stored in the body in the form of triglycerides. As you can see here, this is the structure of the triglyceride. It is known as triglyceride because it consists of the glycerol and the 3 fatty acids are attached with it through ester bond. So that's why it is known as triglycerides. Tri means 3 fatty acids attached by ester bond to the glycerol. Lipid stores are mainly present in A. Liver, B. Brain, C. Muscles, D. Adipose tissues. So lipids mainly stores, and everyone must know that lipids are stored in the adipose tissues, in the adipocytes. So the main site for the storage of lipids are adipocytes, and that makes together the adipose tissues. 8. Glycerol is converted into glycerol 3-phosphate by, as previously now I told you about the triglycerides, in which the glycerol were attached to 3 fatty acids. So whenever the oxidation of fatty acid is required, the fatty acids are removed from the glycerol. So the glycerol, then they are again utilized, and then they can be again enter into the glycolysis. So for glycolysis, they must be converted into, from glycerol must be converted into glycerol 3-phosphate. So the right answer would be A, thiokinase, B, triokinase, C, glycerol kinase, D, all of these. The right option is glycerol kinase, as the name indicates that glycerol is converted into glycerol 3-phosphate with the help of the enzyme glycerol kinase. So kinases mean attachment of phosphate with the help, and this reaction requires the presence of ATP. Glycerol, at third carbon, the phosphate comes from the ATP, which is converted into ATP, and the phosphate attached to the third carbon of the glycerol. So glycerol is converted into glycerol 3-phosphate with the help of the enzyme glycerol kinase. Question number 9. Oxidation of fatty acids occurs in the cytosol, B, in the matrix of mitochondria, C, on inner mitochondrial membrane, D, on the microsomes. So very easy MCQs, as you all must know where does the metabolic reactions or where the oxidation of fatty acids takes place. The oxidation of fatty acids takes place in the matrix of mitochondria. As shown here is the mitochondria, outer membrane, inner the folded membrane, and inside is the matrix. So the oxidation of fatty acids occur inside the matrix. Oxidation of fatty acids requires all the following except A, ATP, B, coenzyme A, C, thiokinase, D, carnitine. They have asked about all the options are correct except one, and that is carnitine. As carnitine is not required for the activation of fatty acids, but it is involved in the transport of large chain fatty acids inside the mitochondrial matrix. So this is the reaction as shown here for the activation of fatty acids. When the fatty acids are activated into acyl-CoA. Acyl-CoA is the activated form of fatty acids, and then after that it undergoes the oxidation of fatty acids. Then it requires the presence of three things: the enzymes, which is the thiokinase, the right option. It requires the presence of ATP and coenzyme A. So all these options are correct. All these ATP, thiokinase and coenzyme A, they are required for the activation of fatty acid, and the activated fatty acid is known as acyl-CoA. While the carnitine is wrong option because carnitine is involved in the transportation of activated fatty acids inside the mitochondrial matrix. MCQ number 11. Mitochondrial thiokinase acts on A, short chain fatty acids, B, medium chain fatty acids, C, long chain fatty acids, D, all of these. So the right option is mitochondrial thiokinase. They only acts on long chain fatty acids, as this reaction is specific for the activation of long chain fatty acids, and fatty acids are activated, and the activated form of fatty acids are known as acyl-CoA, and it requires the presence of thiokinase, ATP and coenzyme A. Carnitine is required for the transport of A, triglycerides out of liver, B, triglycerides into mitochondria, C, short chain fatty acids into mitochondria, D, long chain fatty acids into mitochondria. As we know that carnitine is required for the transportation purposes, and more specifically for the long chain fatty acids into mitochondria. As shown here, the function of the carnitine, as carnitine is required for the transport of fatty acid inside the mitochondria. Carnitine acyl-carnitine translocase is present in A, inner mitochondrial membrane, B, in the mitochondrial matrix, C, on the outer surface of inner mitochondrial membrane, D, on the inner surface of inner mitochondrial membrane. So the right option A is carnitine-shuttle, which is responsible for the transport of activated form of large chain fatty acids inside the mitochondrial membrane. These are present in the inner mitochondrial membrane. As you can see here, this carnitine translocase, it is present in the inner mitochondrial membrane, and they are involved in the transport of fatty acid from the cytoplasm into the mitochondrial matrix. Question number 14. Net ATP generation on complete oxidation of stearic acid is A. 129, B. 131, C. 146, D. 148. The right option is 146. How the stearic acid complete oxidation give rise to 146? Let's calculate how these 146 ATPs are produced. As the stearic acid is a 18 carbon fatty acid. So 18 carbon means that it will give rise to the production of 9 acetyl-CoA, as 18 carbon when divided by 2, as 2 carbons are removed in each pathway in the form of acetyl-CoA, as each carbon are removed or oxidized in the form of acetyl-CoA. So 18 divided by 2, as acetyl-CoA is a 2 carbon molecule, so that's why 18 carbon when they are divided by 2, it gives rise to 9 acetyl-CoA. So 9 acetyl-CoA, it means that the beta oxidation through stearic acid has to go 8 times. So it will follow the pathway, or it goes through the pathway 8 times. So that's why 8 NADH and 8 FADH2 are produced. So 9 acetyl-CoA. So how much ATP are produced? 9 as acetyl-CoA. 1 acetyl-CoA when enters the Krebs cycle, it gives rise to the production of 12 ATP. So 9 acetyl-CoA into 12, collectively it gives 108 ATP production. 8 NADH, 1 NADH give rise to 3 ATP, so 3 multiplied by 8 gives rise to 24 ATP, and similarly 1 FADH2 gives rise to 2 ATP, so it's 16 ATP. So collectively, when we add up all these, and as 2 ATP are required for the activation of stearic acid, so and 2 ATPs will be reduced from it. So total there is the net production of 146 ATP. Again, stearic acid is an 18 carbon molecule which will give rise to 9 acetyl-CoA and will go through 8 times the pathway. So that's why 8 NADH and 8 FADH2. 1 acetyl-CoA give rise to 12 ATP. So the 9 acetyl-CoA will give rise to 108 ATP. Similarly, 1 NADH give rise to 3 ATP. So the 8 will give rise to 24. And 1 FADH2 give rise to 2 ATP. The 8 will give rise to 16. When we add up all of them and 2 are required for the activation, it gives rise to 146 ATP. Propenyl-CoA, formed by oxidation of fatty acids having an odd number of carbon atoms, is converted into A. acetyl-CoA, B. aceto-acetyl-CoA, methyl-malonyl-CoA, D. butyryl-CoA. The right option is propenyl-CoA, which is formed by the oxidation of fatty acids of odd number carbon atoms. Then they are converted into methyl-malonyl-CoA. As you can see here in this pathway, the propenyl-CoA, which will be the end product of the odd number of carbon atoms, as fatty acids are oxidized into acetyl-CoA, but the odd number carbon atoms at the end product, it will be propenyl-CoA, and this propenyl-CoA is again then converted into methyl-malonyl-CoA, and then it gives rise to by the series of reaction, it is converted into succinyl-CoA, and succinyl-CoA, which is the intermediate product of the Krebs cycle, and this is how they enter into the Krebs cycle. Question number 16. Alpha oxidation of fatty acids occur mainly in A. liver, B. brain, C. muscles, D. adipose tissues. The right option is A. liver. Alpha oxidation of fatty acids occurs mainly in the liver and more specifically in the peroxisomes. Refsum's disease results from a defect in the following pathway. A. Alpha oxidation of fatty acids. B. Beta oxidation of fatty acids. C. Gamma oxidation of fatty acids. D. Omega oxidation of fatty acids. So the right option is A. Refsum's disease results from the defect in the alpha oxidation of fatty acids, as it is caused by the defect in the enzyme known as enoyl-CoA-hydroxylase, which is involved in the alpha oxidation of phytanic acid. As shown here in this pathway is the alpha oxidation pathway for the phytanic acid. So phytanic acid, in this pathway there is the defect in the enzyme known as fatty-acyl-CoA-hydroxylase. Because of the defect in this enzyme, it will lead to the result of Refsum's disease. Question number 18. The end product of omega oxidation of fatty acids having an even number of carbon atoms is A. Adipic acid, B. Subbaric acid, C. Both A and B, D. None of these. The right option is both A. Adipic acid and B. Subbaric acid. As shown here, this is the structure of adipic acid and subbaric acid. You can see that it is composed of two carboxyl group on both sides. So this is dicarboxylic acid. So omega oxidation of fatty acids with an even number of carbon atoms can lead to the formation of various dicarboxylic acids. As you can see, it is dicarboxylic acids molecule. So adipic acid and subbaric acid are examples of dicarboxylic acids that can be produced through this pathway. So here are given only two molecules and two examples. So there are many other also dicarboxylic acids. Question number 19. During each cycle of beta oxidation, A. One carbon atom is removed from the carboxyl end of the fatty acid. B. One carbon atom is removed from the methyl end of the fatty acid. C. Two carbon atoms are removed from the carboxyl end of the fatty acid. D. Two carbon atoms are removed from the methyl end of the fatty acids. So the right option is in beta oxidation pathway, it has go to multiple pathways until the complete fatty acid is oxidized. So in beta oxidation, the two carbon atoms are removed from the carboxyl end of the fatty acid. As shown here, the example of the myristoyl-CoA, which is a 14 carbon molecule. So it has to go repeatedly through all these pathways, 4 reactions of the beta oxidation, and in each reaction it gives rise to acetyl-CoA 2 carbon molecule. So 2 carbon are removed, 14 carbon, 2 carbon are removed in the form of acetyl-CoA, then there is 12 carbon, then 2 carbons are removed in the form of acetyl-CoA, then 10 carbon, then 2 carbon are removed in the form of acetyl-CoA, then 8 carbon, then 2 carbons are removed in the form of acetyl-CoA, then 6, and then 4, and then finally the 4 one is divided into 2. The 4 is divided into 2. So 2 acetyl-CoA and 2 acetyl-CoA. The last MCQ is net generation of energy on complete oxidation of palmitic acid is A. 129 ATP, B. 131 ATP, C. 146 ATP, D. 148 ATP. The right option is A. 129 ATP. Let's calculate how the 129 ATP are produced by the oxidation of palmitic acid. As palmitic acid is a 16 carbon molecule, it gives rise to the formation of, it will be oxidized, its carbon will be removed in the form of 8 acetyl-CoA in each pathway. As palmitic acid is 16 carbon, so 16 divided by 2 is 8. So it will give rise to 8 acetyl-CoA, and 8 acetyl-CoA means that it will go through the pathway 7 times. So beta oxidation, there will be 7 cycles. So 7 cycle means there will be the production of 7 FADH2 and 7 NADH. So let's calculate for FADH. For FADH2, 7 multiplied by 2 equals to 14, and for NADH, 1 NADH gives rise to 3 ATP, and there are 7 cycles, so 21 ATP. 8 acetyl-CoA, 1 acetyl-CoA, when it enters into the Krebs cycle, it gives rise to 12 ATP, and there are 8 acetyl-CoA, so 8 acetyl-CoA gives rise to 96 ATP. Let's add up all these, it gives rise to 131 molecules of ATP. and 2 ATP were utilized for the activation, and we will minus the 2 ATP from 131, then it gives rise to 129 production of ATP. That's all for today. I hope you have understood all these MCQs. Practice them, then you will better understand. And don't forget to watch the second part of the video.